Thanks very much, Nick, and colleagues for the invitation. And I have to confess right from the outset that I'm not a bioinformaticist. People in my lab would say I don't know my Perl from my Python from my PHP, and that's true. So we're going to start out, I think, this seminar series with more of a biology focus where I'm trying to get folks interested in, and I've been able to attract some really good people to my lab, to try and understand some of the signatures that we see in the human genome, in the mouse genome mostly, in patterns, and we're particularly interested in DNA methylation. So I want to talk about mostly observational experiments, but I think that will try to get us eventually to the ability to do experiments to test some of these correlative studies. So the fundamental problem that we're interested in, we've spent quite a bit of time on studying projects like ENCODE, or really trying to understand the regulatory events that allow a genome to create different phenotypes, different cell types. And this, half of my lab works in the area of plant biology, and this is obviously a very universal issue. So this is meant for more of a lay audience, but I think it indicates that there is additional information that allows cells to create asymmetries that result in phenotypes like different cell types. Okay, that's kind of the general problem. So this is the most probably other than the microbiome, the epigenome is probably, or epigenetics, the most overhyped word. If you search the news stories about, blame this on your epigenome, blame that on your epigenome. But I'm going to show you a definition that I actually like from Adrian Bird, which really has to do with capturing the idea that there are both molecular events and heritability consequences of having an epigenetic state, and that is structural adaptation in chromosomal regions that signal information, register, or perpetuate the state. So that can either be one cell that divides and maintains that pattern of chromatin or DNA methylation in those daughter cells, or it could be heritable information from even one generation to the other. Now, in the mammalian world, that's quite controversial, although I think there's reasonable evidence for certain types of information. Certainly in worms, there's good evidence for transmission of information. But what I'm going to focus on is the modification that we've been studying, both in plants and people, which is DNA methylation. And so I don't think there's a huge amount of evidence. There is some data that suggests that histones or can be inherited even, some states like heterochromatin states can be inherited from one generation, and I think there's reasonable data for that, but in mammals it's not so clear. But certainly there's data in mammals, and this is the form of methylation that we've been studying, these 5-methyl. There's a hydroxylation of the methyl group to get 5-hydroxy. And then you get further oxidation states, and these are even more rare in terms of their abundance in the genome. But there's good evidence that this kind of modification to DNA can have consequences, and some of those consequences are shown on the next slide here. So if you make mutations in the methyltransferases in the mouse, the mouse dies. If you make mutations in the methyltransferases, you get transposon movement. We see very specific patterns of methylation that are correlated with gene expression. There are some new tools to be able to test whether or not these correlative events are actually causal. There's a beautiful paper in the latest issue of Cell by Rudy Anish, where he's made some tools to be able to add and subtract methylation from genomes. During reprogramming or cell differentiation, you can have dramatic changes in regulatory sequences and their state of methylation. Probably what's most well studied and probably least related to natural biology, though tumors happen, is promoter methylation that occurs during tumor genesis. That's been studied for a long time. Chromosome inactivation, X chromosome inactivation, which has some features that DNA methylation is involved with. And then I think something that's gotten passed over, I think for some time, years ago, decades ago, when I was a graduate student, people would study a specific piece of the promoter, and in fact, individual cytosine methylation events were shown to affect transcription factor interaction, and then people sort of forgot about that. And it's sort of re-emerging, and now you can test in high throughput ways whether or not individual cytosine methylation events have an effect on transcription factor binding, and we and others have been developing assays to do that. And I think once you have that set of data, then you can go back and look and say, okay, these are ones that we believe, at least in an in vitro assay, are affected, and let's look at that in vivo situation for those. So part of it was the absence of having maps of this, either maps from tissues or populations of cells, or even individual cells. And that's sort of where we're going, is from developing methods to be able to read out positional information for individual cytosines. So, you can be fooled by the word, genome-wide. You can have genome-wide and have, one site here and then 100 kb, another site there, another site. It's across the whole genome. But we've been working on developing methods, it's been some time now, for measuring the positions of individual cytosines across the whole genome. And essentially, there's many ways to do this now, but at the time what we do is essentially make a library where the adapters, next-gen library where the adapters are methylated. And then you can carry out chemistry where high temperature and sodium bisulfite will convert cytosine to uracil, except if the cytosine has a methyl group on it. And that's chemistry from the 70s that was developed in Japan, and then applied first by Sue Clark's group to look at DNA methylation in specific contexts. And so that allows you to then do deep sequencing, which gets cheaper and cheaper, although it's still difficult to think about, doing this for personalized medicine. Yes, you can sequence everybody's genome, but as you'll see, different cell types have different methylation patterns. So unless you're really looking for something specific and also are able to access that tissue, it's a bit challenging. So these would be positions where you could map. That's still a little bit challenging for highly repetitive regions, but the length of the sequences is getting better to be able to map those. Then you can quantify the positions of the amount of methylation in different contexts. And you say, well, why would there be a difference, right? You have two chromosomes, and in most organisms, with diploid organisms, you'll have, how can you have a fraction of methylation, right? 50% methylated. So yes, you can have 0, you can have 50% or 100%, but how do you get something in between? And that's mainly because you're dealing with cell populations, and that the cell populations that don't have exactly the same pattern. In fact, if you're having, if the cell's dividing, you're going to have, hemimethylated sequences, et cetera, because the maintenance of methylation is carried out by a specific enzyme. But you can essentially measure the position if you do deep sequencing. You can measure the position, the amount of methylation at any position in the genome. Then you can use a variety of approaches. Some that we've developed, there's a program called MethylPy, developed by Max Schultz, who's now was at HLI, who was, you're able to see, these things you can see, even biologists can see these regions. So these are in fact different tracks here of where gold is CG methylation, and these are IPS cells that have been generated by a variety of methods. This set here, they all have this signature that they remember that in part that they were a fibroblast. It doesn't really matter what method you use to reprogram them. The 4 factors aren't sufficient to reprogram the fibroblast to a perfectly embryonic cell, but they're pretty darn good. It's like 99% of the genome is reprogrammed, but there are signatures that you can see that are heritable, so you can spot those. And then you can cluster that kind of base resolution information across the whole genome. This is for the human, with other kinds of genomic information. The green here is CG. Now we switch colors. The green is CG methylation. The blue is a different kind of methylation, which I'll tell you about in a minute, that we didn't know existed until we had these methods. And then there are other, chromosome accessibility, histone modifications, you name it, to be able to couple that information to get sort of genome-wide maps. And so this is kind of step one, is to be able to catalog information. And you could go to the extreme and say, you know, we want to look at every cell type in the body for this kind of information. In fact, if such a project was just proposed and probably will be funded as part of the Chan Zuckerberg initiative that Steve Wake's group is going to try and develop a cell atlas, at least using RNA data for all cells in the body. And so it allows you to go from assays that capture a fraction of the information to much more, but not all, because of the repetitiveness of the genome, but that with time will be. Actually, a lot of the most interesting stuff is the repeats that are in the human genome or mouse genome. You have to do more targeted ways to attack those regions because of the repetitiveness, but nevertheless, those are really interesting sequences in terms of the expression of genes that are directed by transposons, for example. And there's some nice work from many labs in that area. So I'm going to focus on some neuroscience initiative that we've been working on. I'm not a neuroscientist either, but I'm trying to learn a bit about it from my collaborators. But these are the systems that we work on for different reasons. So I mentioned reprogramming. So that's more or less an ideal system, right, where you can take a cell that's differentiated and reprogram it back to sort of ground state. And there are different ground states depending on whether it's human or mouse. There's sort of a holy grail of creating naive cells, which are really totipotent and not pluripotent, so that they actually make tissues beyond the embryo. They'll make placenta and other tissues. And there are many labs working on that. There's huge controversy in this area. But you have a tool to be able to look at what the in vivo state is in a real totipotent cell that you can get from a mouse or human, and then compare it to what you can make in vitro, and then as you differentiate those tissues to examine whether or not what you've made actually looks like you want to make. And in fact, there is the challenge in regenerative medicine is there's not a single case where you can make a cell that looks like the final cell that you want. So the cells are never really mature in vitro to at least there's no exception that I know, cardiomyocytes and neurons, et cetera, that you really get the final state. So, but you can get information by looking at the differentiation of those cells and looking at their epigenetic state, and you see whether they sort of hit a wall. Right? So you have tools to be able to do that. There are other questions that you can address. Let me go back here. For example, in Arabidopsis, about heritability, we're looking at populations. So we just recently published a paper with a large collaborative group to look at thousands of strains across the globe, collected from all different environments, from more or less desert environments through up into Sweden, and looking at both genetic variants and epigenetic variants of DNA methylation variants in those populations, and looking at the heritability of this information. So you can actually measure over, because the generation time is relatively short, you can measure the generation over 30 generations. You can look at from a single plant through its progeny and look how the DNA methylation patterns diverge over time. So one of the very first things that we spotted when we started developing this approach is that we saw this not in fibroblasts. We saw sort of your generic, off-the-shelf fibroblasts. We saw exactly what was expected, 100% of methylation, where the methyl group sits on a C next to G. When we looked at embryonic stem cells in collaboration with Jamie Thompson's lab in Madison, we found, well, hey, what's this? 25% of all of the cytosine methylation in the genome is in a non-CG context. And actually, Yanish lab had published something a decade earlier about that maybe this was real, and he had looked at embryonic cells and mouse and saw some signature, but Rudy told me, because we're collaborating on lots of projects, nobody really believed that paper gave us. And that's because they didn't really have controls for these experiments, where what was being looked at as a control for whether this experiment worked, that is, did the chemical reaction go to completion, was to use this as a monitor of whether or not you got 100% conversion. So because the religion said you have no C's in a non-CG context, we just look at no C's in a non-CG context on the gene. So I guess a lot of the experiments look like they failed, and in fact that's because this mark existed. And so in embryonic cells, it's quite interesting and different from mostly what I'll tell you, which is But if you look at CG methylation, it does change over development, and I won't go into detail, but not that much in terms of the percentage. But when you look at this non-CG methylation, sort of C next to A is really what's going on here. Is they rank genes based on their maximal expression. Gene 1 here is the most highly expressed. That there's a positive correlation over the, and it turns out this is methylation, and I've removed the gold here, the blue and orange, or the non-CG. Over the gene body, it seems that there's a correlation. And actually, there was recent work from Dirk Schubo's lab that showed that that's because, a very nice study, that there's a mark that's correlated with gene body chromatin modifications called H3K36 trimethyl. And actually, the enzyme that lays down this gene body methylation in embryonic stem cells is an enzyme called DNMT3B. And that actually binds to the H3K36 modification. So I think Dirk has figured out how it gets there, but why it's there is not clear. And that this feature of non-CG methylation is really a hallmark of pluripotency. So you can take any more or less differentiated cell, and it has almost no non-CG methylation. You can add Yamanaka factors to reprogram, and it comes back with the RAGE. And this is compared with two different authentic ES cells. And when you differentiate these cells into something else, it sort of goes away, but we're not really able to get these to mature, so it's hard to know what's going on, and I'll talk about that in a minute. So these are the kinds of studies, I think, that we've done so far, mostly correlative, because we haven't had the tools to be able to, we can have time series experiments that help in trying to determine what happens. So, for example, in these transgenerational experiments, you can see that some genes are completely silenced. That is, they're correlated, the methylation state is correlated with silencing. And then in the next generation, boom, the gene just kicks on. That is, the methylation disappears and it's 100-fold more expressed. And that happens in a completely inbred plant and in the absence of mutation. So there's some variability. And the variability could be just like the machinery for replicating DNA, where it's error-prone. When race makes a mistake, you get a mutation. There could be errors in maintaining the epigenetic state. You have epigenetic events, epialleles we call them, or epimutations that happen. And if they happen to be in genes that affect the phenotype of the organism, you could have something, and it's that's stable. And in fact, I won't show you from the plant world, but there are epimutations that you can't tell. If you do a cross with that, you can't tell if it's a gene mutation or an epiallele. It's stable for the most part, and then you'll see one plant that will throw off this phenotype that's indicative that the epigenetic state was altered. So it's really unclear if this happens in animals or not, but it certainly happens in plants. But today I want to focus on some studies of looking at the most probably complex organ in the body, obviously the brain, with billions of cells, trillions of cells. And there's a lot of interesting phenomena that have been associated with epigenetic changes, and I'll just list a few. So there are many reports that say changes in methylation or in the enzymes that alter methylation, so that put methylation on or remove it, and that's this TET family of enzymes, have been implicated in processes like learning and memory, and also there are correlative studies about the decline of cognition in some mutants that you can make in mice. But a few years ago, there was no such thing as removing methylation from neurons, but it clearly happens now that we can measure it, because those cells are not dividing, and the way that methylation was thought to be removed is that you just don't maintain it. So as a cell divides, you have half the amount of methylation, you have half the amount of methylation. If you don't have reinforcement of the methylation state by this sort of conserved mechanism of DNMT1. But in fact, those cells aren't dividing, and they can be altered in their methylation state, and that's against the sort of dogma as well. So there's that data out there. And so if you can make very specific targeted events where you have deletion of, we've made one recently, and we're still characterizing the mutants, in very specific timing of DNMT3A, which catalyzes this non-CG methylation that I'll tell you about in the forebrain. And this is from other work, but where you knock it out much earlier. Now, knocking it out really early in development actually affects a lot of cell types in the brain. But you see these learning and cognitive defects when you alter methylation. And so one of the things that we're sort of trying to lay the groundwork is that you can do genetics without the genome, but you can certainly do epigenetics without having whole epigenome. But it makes it a heck of a lot easier if you have understanding of the cells that you think are being affected, and you have understanding of patterns of methylation that exist in those cells that you can then test. So a few years ago, we began to test this concept that there are maybe greater changes than what we recognize in the brain. And these are relatively crude experiments where we looked at whole cortex of mouse and human. And that in the cortical tissue, about half of the cells are glia, non-neuronal cells, and the other half are neurons. So we did do some experiments where we purified neurons and separated them from glia in both mouse and human, so that we could track these differences. And so, in fact, this is what we understand of methylation profiles, sort of standard ones. If you're looking at an embryonic cell or a mesenchymal stem cell or a neuron. And then this is the pattern that we see for now removing all the CG in blue, the non-CG methylations. This is that body methylation that I told you about, that you can see that exists in stem cells, but it goes away more or less when you start to differentiate those cells. But this is the amount of non-CG methylation you see in neurons. And in fact, there's more of this flavor of methylation in your neurons than there is of this flavor of methylation in neurons. And just a few years ago, there were all sorts of reasons that this was an artifact, but it turns out that it's absolutely true. And one of the things that we found is that whereas CG methylation, I told you, it actually quite dynamic, and where you see it, if you look at regulatory sequences, as part of the ENCODE project, we've been profiling mouse from day 10.5, embryonic day 10.5 through birth, looking at all the tissues in the body, but particularly the brain. And you see, hundreds of thousands of regions that are dynamic, but the overall level doesn't change much. But the non-CG methylation essentially goes from undetectable to more than what you find in terms of CG methylation, because there are many more Cs. It looks like a smaller percent, right? It's not 80%, it's only a few percent, but a few percent of all Cs is greater than 80% of 30 million CGs. And so you have an accumulation not only in the mouse where we first saw it, but also in human. And this actually turns out to be interesting to neurobiologists, because this is right during a period of where you have synaptogenesis occurring, right? So all the cells are there in the brain, but they're just making their connections. And there's, you could correlate this with anything, with the latest hockey game was or something, right? What the Maple Leafs have been doing over the last decade, right? So, but it's very interesting in terms of something molecular that you can measure that is actually happening in those neurons that are making synapses. There wasn't a lot of information about that. So there's some intriguing hypothesis that we can test by making mouse knockouts, and that's what we're doing for this ENMT3 story. So these are just some of the data. I'll go through reasonably quickly here because it was published a few years ago. This is just measuring, in a purified neuron. This is a cortical neuron, so nu N plus here. These are the nu N minus, which are mostly glial cells. The purple here is the amount of non-CG methylation. So in fact, it's in these are replicates being two different people. That there's an abundance of this context of methylation. And in fact, if you look at this, and this is something that was very surprising, this is not randomly distributed through. If this is just a zoom in, of one part of one chromosome, we're actually looking at base resolution differences. So these are all the possible CH sites. And these are two different people. And these are the positions of the CG methylation in the cortical neurons. And you see that, in fact, it's highly correlated, that two individuals are very correlated. These are three replicates in three different mice. And you can see these at this particular stage of development. There's a very good correlation between the positions. So there's some information that's targeting this non-CG methylation or removing it from other positions in the genome that it shouldn't be, that exists. So there's some machinery there. And these are just showing the correlations between people. Now, the difference between the ES state, now, unfortunately, we've flipped things around, so this is the least expressed gene, this is the most expressed gene, where you have this positive correlation. In neurons, the abundance of non-CG methylation is associated with those genes being repressed. So if you look at this non-CG methylation on a neuronal gene, you can have a very good idea of whether or not that gene is transcribed, just from looking at this data. Now, I'll tell you why we think that is soon. And so you can then cluster these both CG patterns and non-CG patterns and see very interesting features of certain kinds of genes. And so this just catalogs some of the information. So you have constitutively high neuronal genes that during development that are upregulated, okay, and they lose, so they become more blue. They lose gene body methylation, okay, and so here's a purified neuron. You can see it goes from being red here, basically somewhat red, to having complete absence of non-CG methylation or high reduction. And that is associated with the activation of that gene. So you can use these as markers, essentially, to look at either hyper or hypomethylation states throughout development in the brain and have predictions about gene expression that are quite reasonable. So that's published, so I'm going to kind of move through that. So one possible way that this happens was discovered by two labs. There's two papers, one by Huda Zogby's lab and one by Michael Greenberg's lab. Both have been studying this protein, which by definition is a CG binding protein called RETS protein. It's MECP2. And MECP2, in fact, likes to bind better to C in a non-CG context than it does in a CG context. And so genes that are highly enriched in non-CG methylation, and this is just a model of a review that we wrote of these two papers, is the idea that the more the more non-CG methylation you have over a gene body, the more recruitment of MECP2, which attracts repressor complex to this gene. And so, one activity, and there may be others, is that this MC or MCA in different cell types, and particularly in neurons, which have an abundant amount of MECP2, are actually involved in gene repression. How you target that MCA to those particular genes is not clear at this point. And so over development, you start out with, if you look at cells from the inner cell mass or ES cells or create naive cells, they have CA in a CAG context, actually, if you look at that underlying sequence motif. And then as you differentiate them, it sort of it stays CA, but you really don't see a third signature. But when you then start to develop the mature states of later stages in neuronal development after they've stopped dividing, you get a CAC context. So there is some other information that's underlying this motif that may be sequence specific in terms of what proteins it's interacting with. So the challenge for a tissue like the brain, and this is a classical image from Cajal, from a sparse silver staining method, is that there's a huge amount of diversity of cell types. And so one of the things that we've worked on is trying to purify different cell populations and ask the question, is a neuron a neuron, or in fact, do these neurons have different states that you can classify? That you can say, this neuron may have the same electrical pattern, as the neighboring neuron, but in fact it actually knows it has other features that might be involved in its regulation, or where two sister neurons project into the brain may in fact be due to differences in the genes, obviously, that they express, and maybe you can measure that either by looking at the transcripts in individual neurons or looking at the methylation patterns. So just to give you an idea of that, I already mentioned, so in a slice of a cortex, which is shown here, there are layers of the cortex. About half the cells in the cortex are glial cells. The other half are neurons. And of that, 80% of that 50% are excitatory neurons. And the other 20% are inhibitory neurons. And there's really very little known in terms of the molecular differences. So there's a lot known in terms of the electrophysiology. There are fast spiking interneurons that express particular proteins like parvalbumin or other cell types that express other marker proteins that, that folks have used, neuroscience folks have used to differentiate these types of inhibitory neurons. And they also have different shapes. But there's very little known about the kind of molecular biology of those and whether or not you can predict characteristics of different neurons based on looking at the genome in terms of their epigenetic marks. So we collaborated with Jeremy Nathan's lab and Sean Eddy's lab to develop a mouse that had a marker that would allow purification. The concept was developed by Steve Henikoff in principle using different proteins, but nevertheless the same idea, which is there's an integral membrane, nuclear membrane protein called sun proteins that are involved in meiosis. And you can grab this transmembrane domain and make fusions to GFP. And so a student in Jeremy's lab, MD PhD student Alyssa, made a construct that had this SUN1 domain with two times superfold GFP with a MIC tag and target at the ROSA26 locus in the mouse. And this is so-called intact mouse, which you can get from Jackson Labs now. And so what this allows you to do is to have any Cree driver that you want to cross with this mouse that the Cree then, in a cell type specific way, removes this transcriptional roadblock here that has a polydentylation signal that basically removes this. You get expression of the marker GFP, so you can see it. It goes into the nuclear membrane. And so that then Alyssa used three different contexts to drive that expression. She used CAMP kinase, which marks excitatory neurons. And then she used the regulatory sequences for two different classes of interneurons, these fast spiking interneurons called parvalbumin expressing interneurons, and then these VIP neurons. And you can see they're in situ patterns here. These are, as I said, the excitatory neurons are about 50% of the 80% of the 50% of the cells that are neurons. And then these other guys are more like 5 and 1.5%. So small, very difficult to look to measure. And so this mouse then allows you to just basically grind up the tissue, clear it, and then use an antibody to purify cell types that have a Cree-specific expression pattern that's driving that construct. And so these are just some stainings of either GFP or an antibody that recognizes the exact protein that's being expressed. And you can see that these little donuts here that are labeling the nuclear membrane, and that overlaps with, for example, the CAMP kinase signal. You can see the yellow here. And so Alyssa did a lot of characterization of these, and she could show that she had highly specific, that when you purify these are very specific. When you go back and probe these by in situ, you can ask who they are. And the yield is also pretty good. You get about half of the cells that were, in theory there, you recovered about half of them with about 95% specificity. So that allows you to carry out all sorts of assays. Again, these are just snapshots, though, for like, say, one stage in development. So you can purify the nuclei, look at nuclear transcripts, you can look at DNA successibility. So Alyssa did this. Sean Enny's lab did the DNA successibility. We looked at methylation and chromatin was carried out by Alyssa as well. And so then you can look at these things in parallel and ask, okay, here's some of the DNA methylation in the CG context. And so these were whole cortex from fetal stage, whole cortex from the adult stage. And this is now an average of the 50% glia plus the neurons. And so now when you look at the excitatory cells, you can see these discrete, you can see them in here, discrete. These are regions of low methylation, so these are differentially methylated regions that they call. So the interesting part is that, so there's, 250,000 differentially methylated regions that you can find in just these cell types that are relatively small. In fact, you can get this, you can find using different algorithms to get this even smaller. And the vast majority, 95% of these are promoter distal. They're not anywhere near to the promoter. So in fact, the idea that methylation of a promoter is something that regulates the gene is essentially wrong. The only case that happens is in cancer, which is the most well studied. Okay, so you see very little differential methylation at the promoters, maybe a few percent of all the genes. So any change in the edges of the methylation of the promoter. It's all distal enhancers. Well, there's the actual number. I don't know if that's enough or not. And then you can correlate that. Oh, okay, so then when you look at these other rare cell types, you can see they have unique patterns. So these are like 1 and 2% of all the cells. It's very hard to tease that out of the other non-purified populations. So you can find these regions, so what good is that? Well, what good is that points you to regions that are changing in the regulatory regions of those cell types. And then you can overlay that information with, okay, what are the underlying motifs to get an idea of what proteins are binding to those motifs at that particular stage. And so this is the non-CG methylation. I won't go into that right now, I already mentioned that. And then you can look at the chromatin. So here's a TAC-seq assay, and you're looking at, VIP, here's these 3 signals you see in VIP. So there's a nice map that you can get of correlation with open chromatin and DNA methylation by looking at those states as well. Okay, so the system map. So this is what some of the data actually looks like if you're looking at biologists like me looking at a browser, right? So you can find excitatory, so let's say here's a particular gene that has full methylation in this excitatory cells, and here's the 2 inhibitory populations. And here's the cortex if you didn't purify anything, right? It has, or new end, so here's the new end with it, 1 or 2% of these cells are here, right? So you don't see any change in the signal, but there's a huge differential methylated region you get. So maybe this is just all obvious that unless you purify these cells, you really can't see what their underlying state is. And then there's also an interesting observation that was made by Michael Greenberg's lab, is that some of these very specific patterns of methylation, for example, in the VIP and PV cells, and in the neuron, other excitatory neurons, is associated with long genes. So if there's an interesting correlation, I don't really know what to make of it, but long genes have been shown to be enriched. They're long, they could potentially have greater chance of mutations there. They've been associated with lots of genetic events that happen. When you look at GWAS studies for the genes that you come out, often are in these long genes, but these long genes seem to also have an over-enrichment of non-CG methylation. This is what we're looking at here, is non-CG methylation, these long genes. So that's a kind of interesting phenomenon. We're not sure what to make of it. And so, but it's not, if you look at, these differentially methylated regions that occur in these 3 cell types, these 2 inhibitory populations are actually as different from one another as they are from the excitatory cells. That's really surprising. These are sending inhibitory signal to dampen the excitatory output of these cells, but yet they're doing it, at least in terms of their epigenome, very differently. The genes are both transcriptionally, methylation, and chromatin-wise, very different from one another that we weren't able to look at. This just shows some of the correlations that you get that were unexpected. And then, as I said, you can use this information to get ideas about, so in PV neurons, what are the TFs that seem to be enriched at those DMRs? And some of them are surprising. I won't go into the details, but essentially you can pinpoint some genes that we had no idea were involved in those stages of development. And so this is where you can then start testing hypothesis if you're able to alter, for example, the methylation state of particular genes at a developmental stage by some of the tools that Rudolph's lab, Janish's lab has made. Then you can test the idea that there's some causative nature. And so one of the final things that I'll mention here is there are other information that we had no idea really about that existed that is likely to be reflecting what happened at an earlier stage in development of that cell. Okay, so it's a little bit complicated, but Bing Ren's lab actually found that there were regions that surround the promoter, but actually at a big distance, typically 25 kb, around the gene, around the promoter of the gene, where there's never any methylation. They call these DNA methylation valleys. Okay, and those DNA methylation valleys are enriched in transcription factors. okay? So if you just ask who has a DNA methylation valley, there are hundreds of transcription factors that have these. Now, if you look at some of those transcription factors, like this one here, that's involved in development of PV cells, okay? That if you look at the methylation state, the DNA methylation valley shrinks. So you see this collapsing where you now actually have methylation in the DNA methylation valley, only in PV cells. Well, it turns out that if you look at where there's a lot of developmental biology related to transcription factors, that protein is expressed in this called medial embryonic ganglionic eminence. That gene needs to be on at a certain point, and then it needs to be shut off, or else it moves to a different part of the brain. It doesn't end up in the cortex, it ends up in the striatum. And so the hypothesis that we have about this is that those methylation events are to shut off the gene. These are all genes that are regulated by H3K27 trimethyl early in development. They come on, and then instead of having H3K27 resilence them, you may have methylation that then fills in that gap, because you can look through a catalog of, first of all, they're highly enriched in transcription factors, and that, which obviously makes sense because they're DMVs of the transcription factors, but there are hundreds of transcription factors that are linked to these events here that have this H3K27 trimethyl, that there's something known about which cells they're expressed in. And so there's even signatures in the adult cells of what happened earlier in development. And what I didn't tell you is that when we looked through this ENCODE project, where we've mapped out changes in the brain from 10.5 through birth and now after birth, that about 30% of the methylation events that we find, and there's about 1 million events that we can see very easily, about a third of those are linked to regulatory elements. The other 2/3, we have no idea what they are. They're not predicted enhancers. They're not. We don't know whether they're linked to CTCF binding sites or other events. But there's other information that changes dynamically over development that is not linked to known enhancers, unless they're just enhancers that have been missed. But there's been a lot of work trying to catalog enhancers. And so you can use the same tool, and this is another collaboration with Jeremy, to look at other kinds of interesting cell types. Cones are only 2% of all the cells in the retina, and you can see very interesting differentially methylated signals when you use that intact method to study. And this is really what Jeremy's interested in, is retinal biology, or a visual system. And so the final thing is that we want to ask questions about whether or not cells know where they are in the brain. So you have cortical neurons in these 6 layers, and simply going in and using this intact mouse and then dissecting top from bottom, for example, then you can profile these cells and ask, do you know, you're a cortical neuron, but you're in layer 1, 2 versus 5, 6. Okay, and so you can either look at, these are some specific genes, interesting transcription factors, and you can look and say, okay, here's top excitatory neurons in replicate from different mice, and here's other excitatory neurons from the bottom layers, from layers 5, 6, and they're different from one another, both in the CG and the non-CG context, from very interesting sets of genes. So we're really just starting to get at this now, and these are the clustering of those genes. We can find, just looking at top bottom, there's over 100,000 differentially methylated regions, and they're fairly narrowly defined. And so once you get identify those clusters, you can then go to, again, this correlation with, well, what are the underlying binding sites? Do they tell us something? So here's a positive control for transcription factor that's expressed in just the top 1 and 2 layers of the cortex, and that comes out as being enriched in these samples here. And here's one that's known to be in the bottom. You can't see this very well, but these are the bottom layers of the cortex, and that's also enriched. So these are the positive controls. But so it gives you a clue as to what other factors might be involved. The differences can be quite large. So you can see, for example, this is a 415 kilobase region, right, of this GRIN2 gene, which has a very different methylation state in excitatory neurons. And so you can use this as a signature, in fact. If you look at transcripts, okay, this is the amount of space, genomic space that, a percent or so that is gene protein coding or transcribed for protein coding genes. And this is these are the CG regulatory elements. When you look at where the non-CG is in the genome, it encompasses quite a bit more of the genome. So if you wanted to sample from single cells and ask, I want X reads from the genome, and can I place you, using the CA methylation can actually help. You can also use these tools for in vitro studies. So we have a collaboration with Jürgen Knobloch's lab in Austria, where Jürgen's taken individual cells, stem cells, and worked out conditions where you can make little mini brains. And you can look this paper up. It's very cool. You can actually see these little brains that have cortical layers in them. And so we've looked at those, in a collaboration, which is a paper that's under review. And the cortical neurons actually, these are the mini brains, actually show very interesting patterns of methylation that when you look at, for example, what real fetal cortex looks like. So here are embryonic cells, here are these embryoid bodies you can make, and then these are the little mini brains. And so they clustered onto themselves, and they're different from adult brain, but they do start to overlap here with fetal cortex. They actually have some interesting signatures of fetal cortex, and one of the signatures is that same signature that I told you before, the DNA methylation valleys. If you look in those DNA methylation valleys at all the transcription factors, I'll just skip to the final one here. So you have, these are the ES cells, these are the little embryo bodies, these are the mini brains. So the mini brains, and here's real brain, they start to accumulate these hypermethylation signatures in the transcription factors, just like the adult cortex does. And you don't see that in these earlier stages. So they're kind of getting there. They're activating and shutting off transcription factors that would suggest that you're moving towards a real authentic cortical state. So just to wrap things up, I've tried to describe the use of one mark. We're interested in DNA methylation, but there are other kinds of information like histone marks that could be measured. But these regions are changing dynamically, very dynamically through development, particularly the non-CG methylation, which goes from essentially 0 to more than the CG in a matter of weeks in the mouse. Right during this period where you have synaptogenesis, and actually disruption of that, I can kind of give you a precursor, has effects on behavior. And this is a collaboration with my neuroscience colleague, Marga Behrens. And so that's going to be interesting to study those characteristics. But to be able to get at this, we've been purifying different populations using this intact system that Jeremy's lab and Sean's lab developed, and we've collaborated with them, to be able to define the unique patterns of cell subtypes. And I think there's a lot of digging that can go in from folks like you to this data, which is all out there, to try and identify, using different approaches, unique patterns. There are hidden Markov approaches to being able to do this. There's machine learning approaches. We have a random forest approach that we've been using. And there's all sorts of signatures that I think now with some of these new tools can be tested. And ultimately, right, one of the things saying we're piloting this project with doing 10,000 cell types from some of the regions of the brain that we know, but other regions where we're not really sure of, to try and classify cell types, really trying to understand the diversity of the epigenome in both mouse and human brain. And just to wrap up, these are the neuroscientists. So Marga Behrens is a card-carrying neuroscientist. She's fantastic. She's at Salk Institute. And this has been a collaboration with Terry Sanowski's lab. Marga's a staff scientist in that group, but really she runs her own independent group with Terry. Chungyang Luo has basically done most of the experiments I told you on the cortical system, on individual neurons. Yupeng is a bioinformatics graduate student from UCSD who's been working on the ENCODE project, which I didn't really describe. Ryan started a lot of the technology in the lab a number of years ago. And Manoj is a bioinformaticist in the group that's been beginning to integrate a lot of this data with some small RNA data that we now have as well, and then some of the technical staff. Ron is a fantastic computational neuroscientist who is sucked into working on epigenetics of the brain. He has he used to be in Terry's lab. He's now at UCSF, so just across the road, and he's starting a fantastic program of his own. And these are our collaborators, Jeremy and Alyssa, developing Intact with Fred and Lee Henry and Sean Eddy. And just to mention the ENCODE guys, let's get that. I guess I don't have it here. Bing Ren's lab has been leading the effort, and we've been collaborating with him. Lynn and Axel Len Pinocchio and Axel Wissel have been, dissecting all the mouse tissues for the ENCODE project, and I want to acknowledge them for a fantastic collaboration over the years. So I'll stop there. I'm sure I've gone over time, which is my usual phenotype, and happy to answer any questions.
There's a lot there, but at that point you mentioned an example of changing motif context over the course of development, and it was thought to be a non-sequence specific motif. So given that there's now quite a bit known about modification sensitivity and sequence specific motifs, do you think we should reinvestigate putatively non-sequence specific motifs in the context of epigenetic development?
Well, so I know that some factors are highly attracted to a particular motif only if it's methylated. Right, so for Arabidopsis, we've systematically surveyed all 1,500 with success of about 1/3.
Right.
So about 500 or so, about 10% of that 500, it could be a bias set, because it depends on whether it's expressed or not, actually love the methylated motif and don't have much recognition of the one that's not. But 70, the other percentage, some are neutral and some are highly repelled by those. So, I think there is some data in the literature even some from work from, I'm trying to remember, the lab in NIH has done some, so there's also some features of the heterodimer. So if you have, in mammalian systems, you may have a heterodimer, it may be more attractive or not than the homodimer to a protein. So I guess what we're interested in is really saying what, in a kind of relatively high throughput way, and this assay now works, we've tested it for human transcription factors, so we're running through all the human transcription factors on genomic DNA, so that's the thing, you get a context. It's a little harder for the human genome because you've got to get a little more coverage on Arabidopsis. To really get that information and say, okay, let's go back now and test in data like that we have for mouse, what's happening at that locus. Is there a pioneer factor that's attracted, and then, we don't know what, just because there's methylation there doesn't mean it's inhibited. It could actually, in fact, be attracting some proteins. So there's some interesting work from Dirk Schubler. One paper shows very nicely that the factor doesn't care. The next paper, the next factor says, oh, that's critical if there's a methylated residue there. So I think getting that data will allow us to generate some really interesting hypotheses. You can go back with some of the tools that Rudolph's lab and others lab are developing and actually test that idea and say, hey, if we turn on this targeting, this protein to target that locus at this time, does it have an effect?
Right. I guess I was also wondering about time course in terms of things that were not sequence specific but become sequence specific.
Well, if you have we looked for that, we see changes in binding sites that we're at, that there are protein. It's still a big region though, right? So we don't know exactly what's happening for individual motifs, but we know that yes, there's a change in affinity of transcription factors after methylation at a particular site over time, something else, but we don't know how that's related to the exact motifs.
Thanks.
Other questions? Don't be shy.
So what else are you looking at apart from development?
So we are looking at stimulus. So there's some papers that say that you have cortical cells that you can stimulate activity, and that activity causes a change in gene expression, which is true, or methylation state. So we haven't not doing it with cortical with the organoids, but we're doing it with cortical culture explants. So our colleagues have been studying. Take a mouse, E15, take the cortex out, put it in culture, and you can grow it in culture. You can do all this electrophysiology. You can also stimulate those cells with KCL, the Unix amputation of the neurons. And then ask what's happening epigenetically. Well, one thing that we found right off the bat is that those cells behave very differently than you would expect this. Physiologically, they seem okay, but there are thousands of genes that don't methylate in those cortical cultures. And we tried various things like increasing, adascorbic acid, which is a cofactor of TET, et cetera. But you can go in and look at those genes and say, okay, this set of genes have these motifs. Maybe this transcription factor is not expressed at the right time. So these are not they're not developing in the sense that they're dividing mass. They're developing in the sense of maturing to a state, but they're at a critical point where they just stop and they just don't demethylate thousands of enhancers. And those enhancers normally do change in the mouse. And so you can add factors and say, okay, based on the motifs that are there that are not being demethylated, what factors might be the ones that are driving that state? Well, we're doing, looking at projections. So we have a collaborator, Ed Calloway, who's kind of an expert in mapping, using viruses to map different parts of the brain where you're looking at retrograde or anterograde connections. And so Ed's lab, in collaboration with our lab, has been using viruses to label a specific region of the visual cortex in mice where he's giving them visual stimulus that those regions light up, that you can then infect those regions specifically with an AAV, that then those AAV migrate and then infect through one synapse different parts of the brain, and then purify those cells and ask, are the cells that are connected basically by this region, or the target cells the same or different, or are these cells that are projecting to those regions the same or different, because they're very similar looking neurons. So we're trying to really understand the differences in a stimulated neuron of whether or not the epigenetic variation exists in those cells that somehow know to go to different parts of the brain. Or are they just guided, by other signals across cell types that are neighboring and tell them
I have a question. How much of this do you think can be seen on the RNA level instead, right? Because then you're getting closer to the actual phenotype of the cells, what transcription factors they're expressing, what receptors they might be expressing.
Right.
And correlating that, similarly with the connected synapses.
Yeah, so we're
especially the splicing, right, aspect of that.
Yeah, that's challenging. Most of the methods that are measuring single cell RNA are taking a snapshot from one of the ends that's captured on the bead, like dropseq or something. I think that's a very good question. So we know there's additional information in these cells that's not a readout of what's happening in the transcription state. Mm-hmm. And so it could be during the development of those cells, they actually learn to be, that is, they're different as an accumulation of the information across the time. Whereas with transcription, you're just measuring what's happening right now.
Right.
So I think it's complementary. Well, we'll see. But when we get our 10,000 compared to the 10,000, there are some projects measuring 10,000 cortical neurons. But we're actually working on a method to develop getting the methylation and the RNA from the same cell. Now we have some data, and there are a couple of other labs that are doing the same thing. But I think that trajectory across the entirety of development leaves marks on the genome that aren't just a snapshot of what you're doing now. And also trying to get those cells, when you break, when you isolate neurons, you break their connections, and they have phenotypes when you do that. And so looking at nuclear DNA, we know that over time it doesn't change when you disrupt it. If you freeze it or if you let it sit on the bench for 2 days, you don't see changes in the methylation. And so we don't have to worry about the consequences that happen when you alter that. Or if you're looking at nuclear RNA, it's the same thing. There's not much happening. Kun Zhang's lab at UCSD has some nice work on looking at nuclear RNA from neurons, which I think is a better readout of what's happening transcriptionally than looking at cytoplasmic RNA, right? So, but I think it's going to be different information, and the information that either from open chromatin, where you have just basically you don't have a dynamic. There's no you don't have to measure a ratio of this transcript to that transcript for either ataxia or methylation. You have two alleles, you can measure the state, and that. Right, so that it's an interesting question as to looking at the sensitivity. How many reads do you need to see these differences in either chromatin accessibility or RNA to get a phenotype of this cell state or that cell state? Good question.
Well, if you have any more questions, you can join us next door. Joe will be happy to answer those. We've got some pizza and some refreshments next door. Let's thank Joe again for a great presentation.